Hello everyone. So we are going to continue. This is lecture 2 on muscle physiology, where we ended last time. So we are going to continue describing the anatomy. Uh, we are going to continue describing the anatomy of the skeletal muscle before we start talking about the pathways. So what you are seeing right now on the screen is a single muscle fiber, okay? So we're going to look at the structures. So let's start with the first part. We're going to talk about what we call the plasmalemma, okay? So what you are seeing here is a plasmalemma, okay? So this plasmalemma is the cell membrane of a muscle. It's known as a what? A plasmalemma. It has got a very unique name known as plasmalemma. Then what you are seeing here is the what? Nucleus. Then these structures which are greenish, which you are seeing here, they're known as what? Mitochondria. Remember we said that they are multi what? They have got multiple mitochondria. And the nucleus, they have got multiple nucleus. So this is the nucleus, then there's another nucleus there. So meaning they've got more than one nucleus. No, they are saying multiple nucleated, okay? Or multi-nucleated, not multi-nucleated, sorry about that. Multi-nucleated. So they've got a lot of what? Nucleuses. Then let's go inside. You're going to find that they've got fluid, which is found inside, or which is known as what? Sarcoplasm. Okay, not cytoplasm, but sarcoplasm. In the sarcoplasm, that's where you're going to find different structures, such as the mitochondria. Also got what we call the myofibrils. Okay, so these myofibrils, like I said, they contain small structures, or the basic unit of a kidney, basic unit of a what? Of a muscle fiber known as the sarcomere. Okay, then these sarcomere, they are the ones which contract, and they cause the whole muscle to contract as a whole, okay? So that's what you are going to look at right now. Then there's what we call the tubular system, what you are seeing here. Okay, in a skeletal muscle, we have got what we call a triad. The triad involves what? The cisterna and what? The tubular system, or the T tubers. Okay, then what you are seeing here, there's what we call the I band and A band, okay? So these two, the A band and the I band, we'll talk about them later as we go on, okay? But just know that we also have got the A and I band in the myofibril, okay? Then there's what we call the terminal cisternae, and there's also what we call the transverse tubule and the sarcoplasmic reticulum, okay? So when you talk about the sarcoplasmic reticulum, this is an area which stores calcium. Or this is a structure or an organelle found in the skeletal muscle which stores what? Calcium. So all these structures that I've talked about right now, they work hand in hand to perform a particular what? Function, which is contraction of the muscle cell. So key points: the muscle fiber. An individual muscle cell is called a muscle fiber, which I've talked about. A muscle fiber is enclosed by a plasma membrane called the sarcolemma, okay, or plasmalemma. So you can either refer to it as a sarcolemma or plasmalemma. The cytoplasm of a muscle fiber is called sarcoplasm, okay, which contains all the organelles, the mitochondria, sarcoplasmic reticulum, the T tubules, all those things are contained where? In the sarcoplasm. With the sarcoplasm, we have got what we call the T tubules, which allow transport of substances throughout the muscle fiber, and sarcoplasmic reticulum also stores what? Calcium. Let's go to the next slide. Innervation, okay. So meaning skeletal muscles are innervated, meaning they're supplied by the what? They're connected to the nervous system through what? Neurons, okay? So I've got what we call the alpha motor neuron. This alpha motor neuron is the one that transmits chemical substances known as acetylcholine across the synaptic cleft, okay? We'll talk about that later in detail, but let me try to just enlarge and talk about how, or the structure of a neuron looks like, okay? We are still on anatomy. So look at this. We have got what we call the dendrites, okay? So these dendrites are able to pick up signals from what? Adjacent neurons. It might be the interneuron or relay neuron. Then we have got what we call the alpha motor neuron cell body, okay? So this cell body, just like any cell, this is where we find mitochondria, and other type of organelles, okay? Then after that, we are going to have what we call the axon. So this axon, there's what we call, there are two types of neurons that you can talk about, okay? There's what we call myelinated and non-myelinated neurons. So the myelinated one, they transport, or they move the impulses faster as compared to the what? Non-myelinated neuron, okay? So they are going to travel in the axon. So this is direction of propagation of action potential. Okay. So this action potential, if you want to understand what happens here, go and watch the video on what? Cell physiology. Okay, we'll understand that better. So they're going to move down here. They're going to move. So they're going to move here down into the what? The muscle fiber. Okay, until they reach axon terminal, then they are going to reach the motor end plate. In the motor end plate, you're going to find later a diagram which is enlarged, which talks about neuromuscular junction. What happens in the neuromuscular junction? Okay, but right now just know that there is a synapse which is formed between the muscles and what? The motor end plate or the motor neuron. Okay, we'll talk about that later. Let's go to the next slide. So nerve and blood supply. Skeletal muscles are rich in nerves and blood vessels. Chemical connection at synapses, which I've talked about, as neuromuscular junction. Then the synaptic terminal of axon meets motor end plate of muscle cell. Then there's cord capillaries are able to adapt to changes in length of the muscle fiber because muscles are continuously contracting and relaxing. Okay, so here is just a very childish or babish diagram showing a neuromuscular junction, but don't worry, as we go on, I'm going to show a better diagram which shows the pathways involved, okay, and the stages which we so now this slide is just trying to talk about the microanatomy of skeletal muscle fiber, which we have already discussed. Okay, so these are some of the vocabularies that you're supposed to understand in case you are answering a test or you are trying to prepare for a test. These are the different names that you can go through. Just pause the video, then go through them one by one, then you understand what this means or how people can be able to twist these questions. Let's go to the next slide. Yeah. So this diagram again is trying to show the different parts of a muscle cell. Okay, this is a bit detailed, but we can go through it bit by bit. Okay, so one thing you're supposed to understand is that after myofiber here, let me try to zoom out or zoom in. So myofiber, then myofibril, myofilaments, then actin and myosin. So look at this pathway. We start with the myofiber. What's the myofiber? Muscle fiber or muscle cell or myocytes. Then you're going to have myofibril. What's a myofibril? The one that contains the what? The sarcomere, the basic unit of a what? Of a muscle. Then myofilaments. Okay, these myofilaments are made up of two, and they're part of the what? The actin. They're part of the what? Sorry, the sarcomere, which consists of the actin and myosin filament. Okay, the actin filament is a thin filament. The myosin filament is a thick filament, which we'll talk about later. So here, let me zoom out, then zoom in. Okay, let me now zoom out. Okay, you zoom in. Let me zoom in a bit. So now let's look at this. When you look at this diagram, you're going to find that we have got what we call myofibril. Okay, thin filament, then thick filament. Okay, so the thin filament, like I said, involves what? Actin. Then the thick filament involves what? Myosin. Okay, which we'll talk about later. Then they are found in the myofibril. Okay, then these actin and thin filament, they are part of what we call the sarcomere, the basic function unit of a what? Of a muscle. Then when you go, you're going to have the sarcolemma, which I've talked about, the mitochondria, terminal, cisterna, myofibril, sarcoplasm. Okay, then what you are seeing here, this is a tubular system of the what? A muscle fiber. Okay, they are able to transport substances within the what? The muscle cell, and they can also transport things like electrical impulses. Then we have got this. There's what we call, if you hear this word, it's saying a triad. Okay, so a triad involves two things. Okay, it involves, sorry, three things. It involves two cisternae, okay, or you can say two sarcoplasmic reticulum plus the T-tubule or the tubular system. So these three make up a what? A triad. That's what they talk about. Okay, so if you look at the way this diagram is, let me zoom in. We have one here. There's a what? A sarcoplasmic reticulum, which is known as what? The cisternae or terminal cisternae. Then there's also another one here. Then there's what? A tubular system there, the tubular system, which is known as what? A triad. So that is basically called a triad. Sarcomeres, okay? Thick and thin filaments are organized in repeating function units called sarcomeres, okay? Each myofibril has linear arrangement of 10,000 sarcomeres, okay? The banded appearance, which is striation, due to arrangement of thick and thin filaments, okay? So they're trying to explain striated. The point that we skipped on the first slide. So the repeating of this bright and dark filaments, okay, they cause what we call striation, okay. They form more like lines, okay. The thin and thick filaments, okay, the repeated appearance of them, they form what we call striation, okay. Then interaction of thick and thin filaments responsible for skeletal muscle fiber contraction, okay. So when these two, the thin and thick filaments, when they interact, the actin and myosin, when they interact, they cause the muscle to do what? To contract. We'll talk about the mechanism that happens there later as we go on. Let's go to the next slide. Okay. So now, this slide is just trying to show us the structure of the actin filament, okay? So what you are seeing here is the actin filament, okay? Or the thin filament. The actin and the thin filament, they are the same. So let me go through it, okay? So I can't zoom it in because it's very clear. So we have got one, what we call, there's troponin there, there's actin there, and there's tropomyosin, okay? So if you look at these structures, but there's one thing that I've left which passes in between these two actin filaments, okay? This is known as what? Neblin, okay? So neblin is also another protein that passes there. So there are about 4 different types of proteins that make up the actin. There's troponin, okay? There's actin, and there's tropomyosin. Then you have got 2 types of myosin. There's what we call globular myosin and F-myosin, okay? We'll look at that later. Let's go on the next slide. So this diagram is trying to show us another enlarged or zoomed in diagram talking about the what? The thin filament, okay? So when you look here, you are going to find that you have got what we call the sarcomere, okay? So this sarcomere, like I said, is the structure between the starting of one Z line and the ending of another Z line, okay? You can say the beginning of one Z line and the beginning of another Z line, okay? So when you look at this, or the Z disc, so if you look at this, you have got Z disc here and there's another Z disc there. This is known as what? The sarcomere, okay? Then we have got what we call titin. Titin, it's a protein that holds in position the what? The myosin filament. What you are seeing here, which is a bit brownish, is known as what? Myosin. Then we have got what we call the thin filament. We know that the thin filament is what? The actin. The thick filament is what? Myosin. Then there's a line which is found in cutting through the what? The myosin filament. It's known as what? M line. Okay. Then the space between the actin filament and another actin filament, okay, in between is known as what? The H zone. Okay. Then when you look at this part, when you say A band, this is the dark band, okay, from here to there. Then also what we call the light band, from here to there. So the A band involves the what? The thick filaments, okay. That's areas where during striation which appear dark. Then the I band is areas where it appears light, and this is known as the what? The thin filament or actin filament. This is what this diagram is trying to show, okay. There's the Z disc, then there's brightish, then there's dark. So when you look at the I band, it's what? Bright. When you look at the M line, okay, or A band, it's what? Darkish, okay. So that's what they're trying to talk about, okay. I hope you understand it better when you go through the video. Yeah. Thin filament again, or actin filament. So what you're seeing here is another diagram trying to show what? The actin filament. So what you are seeing here, we have got what we call F-actin and G-actin, okay? So G-actin involves of what? A single actin molecule, okay? Then F-actin involves a polymer or a strand of actin when they join up together. Like I said, we have got tropomyosin. Tropomyosin is more like a thread that's bound to the actin. Then there's what we call troponin. Huh? Troponin is more like round structures which bound to the tropomyosin. And this troponin has got three different structures or active sites. Okay, one which responds to tropomyosin, one which responds to calcium, and one which inhibits the binding of what? Calcium. Okay, so there are about three types. Then when you go in, you're going to find that we have got what we call active sites. Okay, we'll talk about that later, but I'm sure you have been able to explain the different structures which the actin filament consists of. So this information is just trying to talk about the different types and the structures or the anatomy of the thin filament, okay, which I've already explained. You can just pause the video and go through these slides. This is what it's also talking about, okay. Then I've mentioned everything, okay. Then let's now talk about the thick filament or myosin, okay, the one which is responsible for showing the dark band, okay. So what you have here, if you can see, there's what we call the sarcomere, okay. We have talked about it, the beginning of one Z line and the beginning of another what? Z line or Z disc, okay. Now we are interested in talking about what? This structure, which is what? The thick filament. So what you are seeing here is a thick filament. Like I said, it's attached to what? Titin. Then when you zoom in, okay, let's zoom in. This is known as a what? Myosin molecule, okay. So this myosin molecule has got this part is the head of the myosin or the cross bridge. You can call it the head of the myosin or the cross bridge. Then you have got the hinge, which is able to move, okay, which allows to have locomotion. Then you have got the myosin tail. So these structures have got different types of what? Proteins involved in. So those are the proteins that we are going to discuss about in the next slide. So this is the structure of what? A myosin, okay? Okay, so you know where the cross bridge is and you know where. So even here, this is where ATP binds, okay? When ATP binds, it's able to make this to move, okay? To undergo movement. Let's go to the next slide. So thick filaments of vertebrate skeletal muscle consist of exclusively of myosin, okay? The length of the molecule is about 140 to 170 nanometers. It consists of a rod-shaped part, okay? When they say rod-shaped part, you know the tail. 2 nanometer thick and 150 nanometer long. And it has a globular part, which is 5 nanometer thick and 10 to 20 nanometer long, which is composed of the 2 subunits, okay? Which we have got what we call LMM and HMM, okay? We'll talk about them as we go on. So the myosin molecule is a hexamer, meaning it has got 6, and it has got a molecular weight of about 470 dalton. It consists of 2 similar heavy chains, which is known as what? Heavy meromyosin, which has got a molecular weight of 200,000 dalton, and the 4 light chains, which are known as what? LMM, which is what? Light meromyosin. Each of molecular weight about 20,000 dalton. The 2 HMM would wound around each other, forming a 2-chain helix, alpha helix. The alpha helix corresponds to the rod-shaped part of the molecule. The rod-shaped part of the molecule. So meaning it's the one that's responsible for moving the what? The rod part of the molecule. Okay. In the amino terminal, which of the two in the amino terminal, each of the 2 HMM forms to form one globular head. The globulars are the SI subunits, whereas the rod-shaped tail of HMM hinge is a S2 subfragment. Each SI subfragment, each S1 subfragments contains 2 LMM, which are of the 2 chemically distinct classes. One of each class is associated with what? Each head. Then HMM of the head carries the ATP activity of myosin. Okay, so there's an enzyme which is able to break down the ATP. Okay, which is known as what? ATPase, okay? So this ATPase is able to break down the what? The ATP when it's bound to the head. And one correction, it's S1, okay, not SI, it's S1 subfragment, okay? There's S1 and S2. So this diagram is just trying to show us what we are talking about: I band, light, which is light band, and then A band, dark band, okay? Then you have the Z line. Okay, so that's the structure of the sarcomere. So this is the end of our second lecture in and these are some of the key points you can look at even these diagrams. Okay, but this marks the end of the second lecture for muscle physiology. Okay, so these are key points you can go through in your own time. Okay.